happy to be here. Like Steve said, I'm an ORISE fellow at the CDC in the Free Living Intestinal Amoebas lab. And I work with Dr. Ibn Ali, who you guys probably, if you've been to this conference before, he has presented. So I joined the CDC in 2018 to work on a new diagnostic test to detect Naegleria fowleri. This diagnostic test is different than others because it's at the point of care. So as we've heard throughout the morning, the CDC receives samples and we provide diagnostic assistance. But ultimately, what we want is to be able to provide faster diagnostics at the patient's point of care. Sorry. Okay, so quick background on diagnostics in the CDC. As most of you probably know by now, there have been 4 survivors in the United States. And one of the key things that has contributed to this has been a quick diagnosis. So these survivors were diagnosed between 2 to 3 days. And how the CDC plays into all of this is the CDC is a national reference lab. This means we provide diagnostic assistance. We are the only national reference labs. However, there are several other labs that exist in the United States that can also help out. So if a clinician or lab personnel ever wants assistance and says, you know, we need confirmation that there is a Naegleria fowleri infection, they will send us the samples and we will process them. And this right here is Dr. Ibn Ali, just for reference. So to go over the diagnostic methods right now, and we've heard about some of them. First and foremost, the front line is microscopy, but as we heard, there are definitely some limitations to this. First of all, that you have to have a pretty high load of the Naegleria fowleri present in the CSF and on the mount to actually see it. Second of all, it may be difficult to discern with other stuff that is present in the CSF as well. And I fully expect that everyone should be able to identify Naegleria at this point with this picture because we've seen it several times. So that can happen at the hospital. At the CDC, there's a couple other diagnostics that we can also help with. So we use ELISA. This is more for environmental samples. This is if we want to track, you know, was Naegleria fowleri present in the water source? So this will kind of help with more environmental tracking and exposure. 2 other things we may use are IHC and IIF assays. So these also have some downsides. We need brain tissue for this, and they take a lot of like technical expertise and special reagents. So the number 1 diagnostic test we use at the CDC would be our PCR assay. And this assay is quicker. We only need CSF fluid for the sample, and we can have results within a few hours. So just to go over PCR super quick, every living organism has DNA, and this DNA is specific for the organism. So when you're looking to do PCR, you're trying to target a specific sequence of the DNA and amplify it. And we amplify it through many cycles, so we have like a large amount, and then you're able to somehow visualize this either through gel or other means. So at the CDC, we have our gold standard PCR, and this is called our Free Living Amoeba Real-Time PCR. It's just another type of PCR method. So this works really great. Like I said, we can have reasonably fast results, like within 3 hours. It can detect really low limits of the amoeba. So if there is one amoeba present in the sample, we can detect it. And it's also specific and reliable, meaning that when we do detect something, we can, when we're providing the diagnostic assistance, we can say, yes, this is Naegleria fowleri. You can go ahead with treatment. So, however, one thing to keep in mind is, although the turnaround time once we have the sample in lab is 3 hours, another limiting factor would be shipment of the sample, just simple logistics. How long does it take the sample to reach our lab? So this is one of the reasons why we need a new diagnostic test. So all of the diagnostics I referenced so far are usually only available in labs. Most of them require really complex DNA extraction steps. They require instrumentation, and they also require data analysis. So these are not suitable in hospital settings. So what we really want to look at is developing a point of care test. Some common point of care tests, we're all pretty familiar with them. So the point of a point of care test is to be able to perform it at the patient or generally in a healthcare setting. So there'd be no need to ship out a sample to the CDC. You can get your answers relatively quick right there in the hospital. So some common ones are like glucometers for diabetic testing, pregnancy tests, and I would even say that the ECG capabilities on your iWatch are also a new type of point of care test. So why LAMP as a point of care? Basically, we want to marry the benefits of a point of care test, something that is easy, accessible, anyone can do, with the robust capabilities of the diagnostic tests we have at the CDC. So if there was some way we could bring the CDC's PCR to the patient's bedside, that would be perfect. And luckily for us, this exists as a LAMP assay. So it has a super complicated name, loop-mediated isothermal amplification assay. However, you should just know that this is like PCR times a million. The end product, you have way more copies of DNA than you would with normal PCR. And some benefits with it over classical PCRs, you don't need a thermocycler. Because it's isothermal, it's amplified at one single temperature. So you can do this in either just a heat block or a water bath. It's a single tube reaction. So once the master mix is in and the sample is in, you close the tube, you run it, and then you'll have your answer. We have designed it with a calorimetric indicator. This means that any sample that's positive will be blue, and any sample that is negative is clear. And this is a picture from an actual run. And like I said before, there's also no special equipment. One of the main benefits is that we have also designed a special DNA extraction method for this that is very simple. It takes 10 minutes. And when you run the assay, you'll have your results within an hour. I designed this nice little infographic to kind of explain the proof of concept of exactly how our LAMP assay will work. So basically, you have your tube of CSF, and with our new DNA extraction method, you will just heat and spin this. So you'll heat it for 10 minutes at 99°C, spin it down, and the supernatant will be your template DNA. The tubes will already contain the master mix needed to amplify the DNA, so you just add your sample, put the tubes in some heat source at 65°C for 1 hour, and then you'll have your answer. And like I said, anything that is blue is positive and clear is negative. So, you know, we spent a lot of time developing this test, and ultimately we all want to know, okay, does it work? Does it work in the real world? Can it even compare to our gold standard qPCR? And so to do this, there are a couple things we want to look at. First of all, what is our limit of detection? And what is the specificity? So the limit of detection is basically going to tell us what is the lowest concentration of amoeba that we can have in a sample that we will be able to detect with this new diagnostic test. And like I said, the CDC gold standard can, if there is one amoeba present in the sample, we will be able to detect it. Same goes for this test. We did a bunch of runs, and basically, to keep it simple, if there is also one amoeba present in the CSF, we will be able to detect it. And the specificity is another important thing. Like we've seen throughout the other presentations, the treatment is pretty robust. You know, there's a lot of drug cocktails, so we want to make sure if we're treating a patient that they definitely have Naegleria fowleri. So we ran several different species of Naegleria and other free-living amoebas. So we tested like this LAMP assay against acanthamoeba, Balamuthia, several Naegleria species. None of them amplified. The only one that did amplify was Naegleria laveniensis. However, this isn't a problem for this test because that is not present in CSF fluid and it's non-pathogenic. That would be another issue if we detect that. So we know that the limit of detection is great, it's just the same as the gold standard, and that we're detecting Naegleria fowleri. So the next thing we wanted to do was test the sensitivity and the specificity again. So At the CDC, we have like a backlog of historic samples from patients that we have already run the gold standard on and we already know the results for. So here, okay, this is wrong. But so we had 9 samples that were positive by the gold standard PCR, right? And when we ran those 9 samples again, they were also positive by the LAMP assay. So that was great. That was like 100% concordance. There were also 17 negative by the real-time PCR. And these were also still negative by the LAMP assay. Sorry, please ignore the false positives and the false negatives. Those were not there. So another thing we wanted to look at, from that batch of samples, one of the samples, we had a series of samples from one patient over the course of their hospitalization. So as you can see in the middle are the days that we received the CSF samples and that we were processing them just to see, okay, as the patient was being treated, what was the detection of Naegleria fowleri in their CSF at that point. The top row is the gold standard real-time PCR, and as you can see on day 3 and 7, the patient showed positive samples in the CSF. On all the other days and towards the end of the patient's stay, there was no Naegleria fowleri present in the CSF. So we ran these same samples with our LAMP assay, and what we found was pretty interesting. So on day 3 and day 7, the samples were positive, just like the gold standard. However, additionally, on days 5, 8, and 9, the samples were positive, which was not shown in the gold standard PCR. This just goes to show that this LAMP assay can even be more sensitive than the gold standard PCR. And in a real-world situation, what this could say is that we may be able to detect the presence of Naegleria fowleri even earlier than the qPCR assay. So overall, the benefits of LAMP is it's time efficient. We've created a crude extraction method that'll make it really easy to run. The results are very easy to read, and because it's portable and inexpensive, this would be great to use in a hospital setting. So all in all, we think we've created a great point of care test that we can start trying to push out to hospitals. So our ongoing work would be our next step. So we've created this and we've tested at the CDC. One thing we want to do now is to evaluate it in a hospital setting. This would include preparing the tubes for shipment. This might include some like lyophilization methods or some sort of freezing method. And then just see how it performs in clinicians' hands or in a different laboratory setting. So with that, I would say it's been great working on this project, and hopefully we'll be able to get this new diagnostic test off the floor and up and running. So I would like to thank the Kyle Cares Foundation, the Smelski Foundation, and the CDC. Thank you. I'll take any questions. Yes. That's true. He asked if the test would work on nasal swabs. We haven't tested that, but probably not. We can discuss it more. Yes.
Had no results. Could you explain why that could have been?
Day 4 had no result. Are you talking about on the gold standard top
Yes.
row? Okay, that meant that it was negative, not that there was no results.
On the bottom row.
On the bottom row?
Yeah, if you could go back to that slide.
Can we pull the PowerPoint back up, please?
Day 3 was positive, day 5, 6, 7.
Mm-hmm. Oh, why that would have been negative?
Why would, yes.
The patient's in treatment, and it could just be, because it detects such a low amount of Naegleria fowleri present in the CSF, it could just be on that day that there just wasn't any present.
Ourania, there was an excellent presentation over here.
Oh, thank you.
I was curious, it looked like maybe it was mislabeled like your box plot was, but it looked like when you went up to 10 to the second, 10 to the third, 10 to the fourth organisms, it turned white again. Was that correct, or was it just mislabeled on your serial dilutions of
Ten-
your?
Yeah, correct. Like 10 to the negative third. So technically, when we made serial dilutions, we could detect down to 0.01 amoebas per reaction, and then after that, we couldn't detect
Oh,
anymore.
that was negative third.
Negative.
It wasn't 10 to the third order.
Correct.
Oh, got it.
Sorry,
Okay.
negative.
Yeah.
Ronnie, Shelley Smelski,
Oh,
Jordan's mom.
hi.
I just have to tell you guys that this is so incredibly awesome, especially for a parent who's lost a child to this. And I can't tell you how amazing this really is, right? It's a game changer. My question is, there is a current case going on right now, we know, in Texas. Are we having an opportunity to leverage this yet on that current case?
I do not think at this time we'd be able to do that. I think we need to go through some more processes. However, I understand, and I think that would be a great opportunity to test it out. And I just want to say thank you for the opportunity to work on this diagnostic test.
Ourania,
Yes.
very nice talk. Can you comment on what is your gene target that you're amplifying for, and how much diversity do we know about that in Naegleria fowleri isolates?
So we were targeting the mitochondrial DNA sequence of Naegleria fowleri, which has several thousand copies present in the cell. What was the question?
Yeah, so you said there was an overlap with lovinensis. Is there a specific sequence of, is it cytochrome B, cytochrome oxidase 1 that you're actually amplifying, and wondering how much difference there are among any other isolates of fowleri?
I see. I'll have to get back to you about that. Yes.
Hi. How much more time does it save? Like, does it, I guess, save a day of pain in a way? Or like how much, what's the ratio, I guess, between a regular diagnosis and your test?
So I would say if it's present in microscopy, that's great. You can start treatment right away. If it's sent to the CDC, usually we try to have this back. So like I said, depending on shipment and logistics in that sense, by the time we receive the sample, we can get the diagnostic results back in 3 hours. That's usually 24 to 48 hours.
Wow.
Right. So in this case, if we could do it at the hospital, it would save potentially 1 to 2 days diagnostic time. Mm-hmm. Yes.
Can you get an idea about the genotype of Naegleria fowleri from your test? Because we know that there is drug susceptibility difference among genotypes.
Right. We were able to detect all the genotypes with this test. We were running that with the specificity testing. Yes.
Because this is a point of care test and probably more sensitive than microscopy, this and easier to do than microscopy, and results in an hour. This is like a great opportunity for improving diagnosis of PAM compared to what we struggle with now.
I totally agree. And I think at the beginning when we're starting off evaluating this, if we're doing microscopy and we're unsure, like Sheila was saying, she had like an inkling that it may be a PAM case, why not just run this test as well? Like I said, it's also affordable, so it does no harm to just run this along with your normal testing. Yes. So the tubes, correct. We would create. Go ahead.
but in terms of equipment,
Equipment, yeah. So we want to prepare this test so that we can ship it out and the tubes would be ready to go. This would have your positive control, your negative control, and all tubes for your actual template DNA. The only equipment a hospital would need would be anything that I think would be in a standard laboratory, a set of pipettes, and a heat block or a water bath. That's it. And a centrifuge. Yes.
So you had said that you needed a special DNA extraction method. Is
Yeah.
that something that would then have to be routinely implemented into those specific diagnostic labs in every hospital?
No, if there is a DNA extraction method that the lab is more comfortable with, for instance, we also extracted the DNA with, I think, just a normal QIAGEN kit. However, this heat and spin method is just so much easier. So extraction kits are expensive, as we all know. The reagents, they require kind of like some technical expertise. This method, you would use the same equipment that you would use to run the lamp. If you have a heat block, you simply just aliquot out some CSF, heat it, and then you spin it down in a centrifuge, and then that supernatant will contain the DNA that you would need. But no, they don't have to use it. However, I think it would significantly cut down on the time as well, and cost. Yes.
Yes, this is really exciting. Great work.
Thank you.
My question was, does this require FDA approval? And if so, how long would that process take?
Okay, that's a good question. I'm unsure about that, but I'll definitely look into it and get back to you. Oh, that was your question too. He asked if this was ready for prime time, like if it's ready to go. So everything that we've done at this point has been in-house at the CDC. So, that's in our hands. We're used to dealing with Naegleria fowleri, and so are hospital lab settings. However, as we all know, tests are different in different people's hands, different collection methods, different DNA extraction methods. So we really want to have like clinicians and other lab personnel test this out, and that would be the next step. How does it begin? I think that we would start contacting certain clinicians, probably Florida clinicians too, because there's interest here to have them, if they have their own stock of Naegleria fowleri, they could just test it like in vitro. If they wanted to test some with CSF, that's fine as well. So we would need to make some contacts and get it out there to try.
Health, just as a pilot place, could be an option, you're saying?
Yeah, that would be an option.
Okay.
Yes.
If it's not FDA approved, we can always have it as an LDT. And we're very excited. So I really hope that Advent Health will kind of piggyback onto this. If you send us the standardized kits,
Mm-hmm.
I am sure the heroes to be hopefully are here listening
Right.
because we want it.
I think that would be a good idea too. Any more questions? You can always come talk to me after too about this. I'd be happy to discuss it.